All right, I wanted to go over some of the brain structures with you. People wanted videos, so we're going to make some more videos here for you. We have both mammal brains, we have shark brains, we have some other species. And the idea, right, is to show the comparative anatomy amongst all of these different species when it comes to the brain and when it comes to cranial nerves. For the most part, one of the big take-home messages is that things stay relatively the same. Whether we're dealing with the lamprey, or we're dealing with a fish, sharks, mammals, whatever it might be, reptiles, birds, amphibians, all of these species are going to have 5 different regions to their brains, and they're all going to have the same cranial nerves. The cranial nerves may change names. We might add some cranial nerves as we move through our phylogenetic tree, but for the most part, everything stays relatively conserved throughout evolution. We can jump first to our shark brain. The first region that we would have would be the telencephalon. Following the telencephalon, there's this region here connecting the two. This would be the diencephalon. Behind the diencephalon, you would have the mesencephalon, the medencephalon, and then the myelencephalon. Remember, if you're having trouble, mesen, meden, myelin, it's in alphabetical order: mesen, meden, myelencephalon. So there are 5 regions to the brain, and that would stay true if we're dealing with our bony fish. Over here's the medencephalon, here was the mesencephalon, diencephalon, the telencephalon's up within there, there's some brain. Oops, falling apart. But it would stay true with our fish. We can't really see it too well with our lamprey, but following this olfactory sac, here would be the brain, and it would have those 5 sections as well. I'm never going to have you identify things on the fish or the lamprey, but I want you to realize that it would hold true as well. As we go through, the fish and the lamprey would have the same regions and cranial nerves as the shark would. Okay. If we go to our mammal has a telencephalon. Telencephalon. Telencephalon would be up in here. Telencephalon has become folded back. So all of this is folded back on the other regions. It's just become greatly enlarged. We have a diencephalon. We have a mesencephalon. We have our medencephalon, which comes up to the front here and includes this region as well. And then we have our myelencephalon. I have to struggle with this light a little bit here. Make sure you can see everything. And I'll go through those again. So telencephalon, diencephalon, mesencephalon, medencephalon, and myelencephalon. We'll go through those slowly throughout this video. So one of the things I'm going to do is I'm going to go through these different regions. I'm going to talk about what those different regions do, the primary functions. There's a lot of functions amongst all of these different things. We're simplifying things for the sake of a one section of our long lab. But if you were to go into a neurobiology class or even a major, you would learn all of the different functions going on within these different regions. I'll primarily start with the shark, and then we'll bounce back and forth between the shark and the mammal. So if we start with the telencephalon, here's my telencephalon. Okay. The telencephalon, if I shaved off some of this more superficial region and I go deeper, you'll see there's a nerve coming off of the telencephalon. That nerve goes to a bulb, that bulb goes to a sac, and that sac sits right by the nares, where water is coming in and out. That's where the shark is smelling. Chemical information is brought into that nares. There's nerves, small neurons within this olfactory sac that connect to the olfactory bulb. that transmit that sensory information of olfaction, smell, chemical information, chemical processing, up to the telencephalon. The telencephalon's primary function for many of these different species is smell, processing sensory information of smell. Now notice that the brain is very linear in this species. Telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon. And that holds true for many of these other vertebrates that we looked at. These are called linear brains. And for the most part, when we have these linear brains, the telencephalon's primary function is olfaction, smell. It's not till we get to the mammal that we see that that telencephalon has folded back now on these other sections. Telencephalon has folded back on these other sections, but still you have your olfactory bulb transmitting to the olfactory tract, olfactory nerve, going to the telencephalon. You can see that here. Let's show a good one. Here's our full brain olfactory bulb on each side, olfactory tract, going to our telencephalon. The other function of our telencephalon, once again, here's my telencephalon. Here I have not shaved off that superficial areas of the contracranium, so you cannot see the olfactory tract. Here you can see still the telencephalon. We also refer to these regions of the telencephalon as the cerebrum. These are my two cerebral hemispheres. And the other function here of my telencephalon / cerebrum is processing information. This is where a lot of neuronal connectivity is occurring, cause and effect. You form connections here from neuron to neuron, so that as you bring in sensory information to the brain from the rest of the body, you have a cause and effect relationship. This sensory information comes in and it causes this effect going out. This is also what you might be referring to as logic or thinking. And as we move to our terrestrial animals, and in particular our mammal, we see a great enlargement. Try to make it so there's less glare. We see a great enlargement of the cerebrum. Hey, that works. This greater enlargement of the cerebrum allows for more neuronal connectivities, more thinking, more logic, more processing, especially when you're in this larger, more open three-dimensional space that is terrestrial environments. So one of the big differences we see with mammals is a great enlargement of the cerebrum, a folding back of the cerebrum on the rest of these sections of the brain, these regions of the brain. That does it for the telencephalon. The telencephalon, we have the one cranial nerve, the olfactory nerve. We have smelling brains, the processing of smelling information. And then we also have thinking, logic, these connections, processing all the information coming into the brain and sending out reactions. The next section we have is the diencephalon, the next region. Sorry, when I say section, that you can replace that with region. This sunken area here between these two bulbs, this is the diencephalon. The diencephalon is typically covered by a thin membrane sheath. You can see on this one, it hasn't been removed. That membrane sheath is referred to as the telecordia. On the other side of the telecordia would be the choroid plexus. If I look at a mammal brain, I cannot see that there's no like thin membrane sheath within here, but this would be my choroid plexus. Notice when I pull on this, you can see there's a space, a gap within the brain here. Choroid plexus lies on the anterior section of that gap. The choroid plexus primary function is production of cerebral spinal fluid. That cerebral spinal fluid will fill these gaps. These are called ventricles. The cerebral spinal fluid will then come out through what's called an aqueduct and now bathe the brain in an osmotic, balanced solution, allowing for perfect environment for neuronal activity. That cerebral spinal fluid will also bathe the rest of the brain here, go down the brain stem, pass out the foramen magnum, and bathe the entire spinal cord. So on my shark, you can see, come out here, bathe the brain, and pass down, bathing the spinal cord. There's a lot of other things going on within the brain here. I'm going to use the mammal brain just because the diencephalon is a lot larger. This whole entire region here is the diencephalon. The diencephalon is going to be composed of the hypothalamus and the thalamus. The thalamus will be more dorsal to the hypothalamus being on the ventral portion of the brain. The primary function of the hypothalamus is to control the pituitary gland. Let's see. The pituitary gland gets ripped off the majority of the time. You can see something has been ripped off right about here. That would be the pituitary gland in particular. The primary function of the pituitary gland is to release hormones. There's a lot of hormones that are being released by the pituitary gland. Many of those hormones are going to go and affect other glands within the body, the thyroid gland, the gonads, the thymus, the adrenal glands. Many of these then release their own hormones that have effects on things within the body, helping the body function and respond to particular stimuli within the environment or as the organism develops. The pituitary gland is releasing those hormones that help regulate how the body's functioning, or goes and affects other hormones. The pituitary gland is controlled by the hypothalamus. The hypothalamus can be referred to as like the puppeteer, and the pituitary gland being the puppet. Okay. The thalamus, the more dorsal region, is primarily going to be affecting, it's like a relay center. Sensory information coming in from the body, or some of these cranial nerves, will pass through the thalamus, and the thalamus will then relay that sensory information to different regions, like the cerebrum, for further processing. Another gland that we have coming off of the diencephalon is this one here. That little peanut is the pineal gland. Notice if I tuck back in there, you can see the pineal gland deep in there. On these linear brains, like the shark brain, the pineal gland would be right back in here along this dorsal section of the diencephalon. Remember, that is covered by a thin membrane sheath, like what we saw on this shark brain, that's fairly translucent, just as the chondrocranium would be. Light can directly impact the pineal gland on these linear brains. In fact, some reptiles have a small hole sitting on top of their skull, allowing for light to easily pass through and affect the pineal gland. The pineal gland is going to help regulate melatonin, which is your sleep-wake cycles. So when we deal with a mammal brain, we're going to run into problems because now this enlargement of the cerebrum, or the telencephalon, is directly covering up that pineal gland, so light can no longer impact it. We're going to have to find a way around that. The diencephalon and the telencephalon are the forebrain. Let's move now to the midbrain. So now we're within this section here, these next two lobes. This was the telencephalon, diencephalon, mesencephalon. On my mammal brain, the mesencephalon is all within this region. The mesencephalon is going to be bringing in sensory information from the eyes, and it's also going to be responding back to the eyes with motor information, movement of the eye. The first big nerve that we have here is the optic nerve. So the optic nerve would be coming in here and on this side, and you can see that it crisscrosses right here at what's called the optic chiasm. Typically those optic nerves get pulled off, so I don't really have a good example of how they're coming in. You would have one coming in here the other one coming in here, and they crisscross at the optic chiasm. That's the optic nerve. The optic nerve is bringing in sensory information from the eye. Fun little show. I have on this brain here, so this is the ventral side. I've dissected out the brain from inside the oral cavity, and you can see this was my telencephalon, my diencephalon, telencephalon, diencephalon. Here's my mesencephalon, and this right here, coming off the bottom, is my optic nerve. Let's see if I can't give you a good view of that. I don't know if this will be too close. But there's my optic nerve. So just like on the mammal, the optic nerve on the shark is bringing in that sensory information from the eye. That's coming into the mesencephalon where it's being processed, and then reactions can come about from there. The next nerve that we have, let's see if I can't find it here. It gets ripped off sometimes. Be a small nerve off to the side. Looks like it may have been ripped off these. That would be the trochlear nerve. Right here is the trochlear nerve. So here's my optic. Here's my trophic. Oh, I'm sorry, I skipped one. Olfactory, optic. This big one is the next one. That's the ocular motor. After the ocular motor is the trochlear nerve. Okay. Both the ocular motor and the trochlear nerve are going to be sending motor information back out to the eye, so moving the eye. Okay. There'll be one more cranial nerve that goes and does motor to the eye. We'll deal with that one in a little bit. I have a nice little model of a human brain here. So you can see we have the olfactory nerve, the optic nerve. This would be the optic chiasm. This would be the pituitary gland. Here is your ocular motor. And off to the side is your trochlear. Try to show that in a better light. Reduce glare. This might be the best. Olfactory, optic, ocular motor, and then you have your trochlear. Okay. Okay. We can keep trying to move that light around to get the perfect view without getting too much glare. Okay, and that's it. That's it for the mesencephalon. We did optic nerve, optic chiasm, trochlear, and ocular motor. I have on your handouts optic lobes, tectum, tegmentum, optic tectum. Get rid of all of that. We're not going to go that far into depth. Okay, the next thing is going back to that optic chiasm. So let me grab my mammal brain. Going back to the optic chiasm, we had that crisscrossing occurring there. The one thing I want you to know about the optic chiasm is we have a specialized structure in there. It's called a clock gene. In particular, it's the suprachiasmatic nuclei. You have many clock genes throughout the body. Clock genes are very special. What they do is they produce a product, a protein, that then goes on and has an effect on something. All right? The neat thing, though, is that product that goes and affects something down the road also can have feedback on the clock gene itself. So that this suprachiasmatic nuclei, as it's producing a product, that product comes back and affects the suprachiasmatic nuclei. Such that, as that product increases in concentration, it shuts the clock gene off. As the concentration of that protein begins to fall, the clock gene then turns back on. And as the clock gene turns back on and starts producing the product, and the product increases in concentration, it turns the clock gene back off. And so as the product increases and decreases in concentration, you could imagine it's going to create this rhythm of whatever it's going on to affect, because it's turning on and turning off, turning on and turning off. And that rhythm could be a daily rhythm, it could be a monthly rhythm, a weekly rhythm, a yearly rhythm, it could be even every 2 years, whatever it might be. And so this can create circadian rhythms within the body, or circannual, whatever it might be. This one in particular is going on to affect the pituitary gland. Remember, we had that issue with the pituitary gland being hidden away. Now we have a way for light to affect the pituitary gland. The rhythm created by the suprachiasmatic nuclei, it can be directly impacted by light, visual information, there in the optic chiasm, so that now not only do we have a rhythm of melatonin production, but that rhythm can then be entrained by light information coming in from the optic nerves. So you have a rhythm within your body of melatonin. If you were to be locked away in a dark closet for a month, you would maintain that rhythm of melatonin. If you were to go and travel to Europe or Australia, somewhere on the other side of the world that might have, a different light rhythm going on, because you're traveling away from where we are, you're going to have jet lag, you're going to get tired at the wrong times, but as light information comes in and affects that suprachiasmatic nuclei, it's going to affect that clock gene. And then it's going to impact your melatonin rhythms and your melatonin productions and get you back on your right cycles. This is extremely important for organisms that are interacting with their environments and that need to maintain activities at certain times of the day and not others. Melatonin is going to be what's helping entrain that, and the direct effect of light on the pineal gland or on the suprachiasmatic nuclei is going to help entrain those rhythms. All right, moving on. So now we're done with the mesencephalon. We can now move to the medencephalon. So here is the mesencephalon. Here's the medencephalon. The medencephalon in the mammal, you can see here. It wraps around, and it also is this structure back here. The mesencephalon is technically the beginning of the medulla oblongata. This is the medencephalon. The medencephalon is technically the beginning of the medulla oblongata. But for the most part, we're not going to talk about the medulla oblongata here. We'll save that for the next section, where the majority of the medulla oblongata is. The big thing I want you to get from the medencephalon is this is where we find the cerebellum. This is technically, this lobe on the top of the shark's brain is the cerebellum. On the mammal brain, this is the cerebellum. As you can see here, this cross section, this is the cerebellum. The cerebellum has two main functions that we're going to focus on. The first is processing fine motor control. If you damage the cerebellum, you can still move around, but you're going to be moving in a very uncoordinated pattern. You're going to be very jerky movements. You might be tripping a lot. You might not be able to grab things very nicely. Your fine motor control is going to be gone. The other big thing that we're going to focus on is three-dimensional mapping or memory. And so if we were to damage our cerebellum, not only are we going to be moving in uncoordinated fashions, but we're also going to have trouble finding our way home. So remembering where things were within our life. That could be extremely important for organisms that are interacting with their environments. Okay. There are no cranial nerves coming off of the medencephalon. The rest of our cranial nerves are coming off of the myelencephalon. So let's move now to the myelencephalon. The myelencephalon is the rest of this medulla oblongata. In our shark, you can see it really nicely here. This would be the myelencephalon. Okay. The myelencephalon is an extension of the spinal cord as it runs up into the cranium. Now that we're up within the cranium, we're going to refer to this as part of the brain, and this would be the myelencephalon. All of your large neurons within your body are processing sensor or are moving electrical information down those long axons, And a lot of that electrical information can be lost from the axon if it wasn't insulated. So we insulate our large axons with myelin sheaths. Those myelin sheaths will continue up into the brain here from the spinal cord and along this posterior section of the brain. And so that's why it's called the myelencephalon, because there's myelin sheaths still retained within this portion of the brain. The medulla oblongata, you can think of the primary function being like a road for all of this information, this neuronal information coming in from the rest of the body to the brain to be processed and then sent back. And same with the cranial nerves. You have a large number of cranial nerves coming in at this region as well. So let's go through those cranial nerves. The first one that we have, let's show you on the model here first. So here was my olfactory, optic, ocular motor. Off to the side was my trochlear. And then back here, this large one is the trigeminal. I go to my mammal brain. It's typically ripped off. We can't see it well. Olfactory, optic, ocular motor. Trochlear was off to the side. And then trigeminal would be somewhere around here coming off. But it's been ripped off. If I look on my shark, my trigeminal would be right around here. You can see there's kind of this swelling. Right below here would be the trigeminal. I can't show you on this. Okay. If I were to show you on the handout that you have, this would be your trigeminal. The trigeminal is going to split into 3 branches, but it's 4 actually on most other vertebrates aside from mammals. The 3 branches will be the ophthalmic, then you would have the maxillary, and then mandibular. Okay. The ophthalmic in the shark will be both the superficial ophthalmic and the deep ophthalmic. And you can actually see those really nicely on our sharks here. This is the superficial ophthalmic. You can see running up on both sides. It runs behind the orbit. And then you can see it being superficial right here. If you go to the optometrist, you're looking at your eyes. The ophthalmic goes to the facial muscles around the eyes. And the primary function is going to be picking up sensory information from those facial muscles. The deep ophthalmic sits along the back of the eye. You can see it right there. It's right here, along the back of the eye. And the deep ophthalmic is doing the same thing. It's picking up on sensory information from muscles on the face, near the eye, but obviously this one's running a little deep. We then would have 2 more. I can't show you too well on these. You would have the mandibular and the maxillary branches. The mandibular is going to the facial muscles on the mandible, or the shark, that would be the Meckel's cartilage region. And the maxillary is going to where you can imagine the maxillary and premaxillary bones, so your upper lip for the most part. Would be on both the mammal and on the shark. Okay. And all of that is sensory information of those facial muscles. Now, once again, we're kind of simplifying things. So if you were to go in more depth within your textbook or within a neurobiology course, there is some motor control that's going on with the trigeminal. For example, mastication, the masseter muscle, motor control of the masseter muscle is controlled by the trigeminal. But I'm going to simplify things for you. I don't want you to go too far in depth. We'll just say sensory of facial muscles. Okay. The next one we have, so now what we've done is we've gone to olfactory, optic, ocular motor, trochlear, trigeminal. Now we're going to jump back to the middle of the medulla oblongata, and we're just going to work our way around. Okay. And so for this part here, we're going to go A, F, A, G, V, A, H. That's for the mammal. For the shark, we leave off those last 2 because the shark doesn't have those last 2 cranial nerves. The next one we have is abducens. If I flip this around, see if we can get a better view. Now you can kind of see without that glare. Here's that optic nerve. If I go a little further back in here, haven't quite uncovered it yet. But deep in here would be the abducens. The abducens is on the posterior side. Actually, here it is. It's right deep in there. No, that's chondrocranium. You can kind of see it coming off. You probably won't be able to see it. But it's deep within here, coming off the medulla oblongata is the abducens. Let me show you on the big brain here. We've done olfactory, optic, ocular motor. Then we went off to the side, trochlear, trigeminal. We came back to the middle. We're going to do A, F, A, G, V, A, H. The first one right here in the middle is abducens. Now remember, we did sensory of the eye, motor of the eye, motor of the eye. Let's finish off motor of the eye. Abducens is going to abduct the eye. It's going to have the eye look away from the midline. Okay, abduct the eye. That would be abducens. So that's motor of the eye. Now we're done with the eye. After A, we go to F. F is going to stand for facial. We've already gone to the facial muscles. Remember with the trigeminal, that was sensory. This is going to do motor of facial muscles. Motor of facial muscles. A, F, A. This next one is A. It's a placeholder, auditory. It's going to be called two different things depending upon what species we're looking at. If we're looking at the mammal, it's going to be the vestibular cochlear nerve. If we're looking at the shark, it's going to be passing back to the sacculus and the semicircular canals. That's picking up on auditory sensory information and equilibrium sensory information. So it's the statoacoustic in the shark, and it's the vestibular cochlear within the mammal. All right, coming back to my model. A, F, A. This one should be white. They have it brown. This would be G. This is glossopharyngeal. Glossopharyngeal, you say glossal, I say tongue. You say pharyngeal, pharynx, I say pharynx. Okay. This is going to be going and doing sensory of the tongue. Okay. It's also going to be doing sensory and motor of the pharynx region. So all of these muscles that are controlling the branchial constrictor muscles, right, controlling opening and closing these branchial pouches, all of that is the sensory information. If you punch a shark in the pharyngeal slits, it's going to feel that via the glossopharyngeal, and it's going to then also respond motor movement via the glossopharyngeal as well. So this is sensory and motor of the pharynx, and this is sensory of the tongue. Let's see, actually, I think one of these, you can actually see the glossopharyngeal right here. It's hard to show on the video because of the glare. But so we have, here's my sacculus, and the glossopharyngeal is passing, going back to that pharynx region. Going back to my model. A, F, A, G, V. This big one here is V. That's the vagus. On my shark, you can see that one is the most posterior cranial nerve coming off. It's the vagus. And this one here, you can see it back in here. That's my vagus. Okay, glossopharyngeal would be back here. The vagus is going to go and innervate many things throughout the body cavity, many of the organs throughout the body cavity. Just pick your favorite. In particular, it's going to be innervating the heart. Then it's going to pass down more towards the abdominal cavity in mammals and further in the body cavity in sharks, and it's going to be innervating things like the stomach, the liver, the pancreas, the intestines. It's going to innervate all of those things. It's going to do sensory and motor. Okay, and just pick your favorite organ down there in the body cavity. In the mammal, it's going to be innervating the lungs as well. Okay. Vagus comes from vagabond. Vagabond means somebody who wanders. That's it for the shark, but if we continue with the mammal now, we have two more cranial nerves coming off the medulla oblongata, the myelencephalon. So we had A, F, A, G, V, A. This is the accessory. Now if we go back to my shark here, the shark, once we leave the foramen magnum, we go down the spinal cord, we're going to have a lot of spinal nerves that go out of those neuronal foramina, going out to different sections of the shark's muscles. Same with the lamprey. Same with any of these other organisms we looked at, cephalochordata, amphioxus, whatever it might be. They all have spinal cords going down that dorsal hollow nerve cord, right? Or that dorsal hollow canal. And the spinal cord will have spinal nerves going off. You can almost see them when I kind of stretch this away. Little spinal nerves going out to those skeletal muscles. That's going to be doing sensory and motor of those skeletal muscles. And we've learned about a lot of them. On the mammal, one of those spinal nerves has basically just moved up into the brain case here. And now that it's within the brain case, the cranium, we're going to refer to as a cranial nerve. So this accessory nerve was simply just a spinal nerve before. That's now a cranial nerve because it's within the cranium. But it's going to be doing the same thing as it did before. It's going to be doing both sensory and motor of some muscles. It's going to be doing sensory and motor of the trapezius complex and the sternomastoid. Remember, the sternomastoid is the sternocleidomastoid when we're dealing with humans. So if you're familiar with human anatomy. That same nerve is here in the shark. It's just down in the spinal column. And it's going to be going to the homologous muscle of the trapezius complex and the sternomastoid. That would be the cucularis. So the shark doesn't have that cranial nerve, but it still has that nerve. It's just a spinal nerve. Going back to my mammal, after the accessory, come back up to the middle, I have H, hypoglossal. Remember glossal, tongue? We've already done tongue, but we did sensory of the tongue. This is going to do motor of the tongue, movement of the tongue. Sharks don't have a true tongue. They're not going to move their tongue around. They don't have those muscles we talked about, like the hypoglossal, styloglossal, all the genioglossus. All of those are moving the tongue around. Those are going to be innervated by the hypoglossal. Okay. So just to recap here, we have my cranial nerves, O, 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 T, T, A, F, A, G, V, A, H. Olfactory, optic, ocular motor, trochlear, trigeminal, abducens, facial, auditory, which is a placeholder, it's vestibular-cochlear in the mammal, statoacoustic in the shark, glossopharyngeal, vagus, accessory, hypoglossal. Sharks do not have a hypoglossal. They don't have an accessory, but they do have that nerve. It's just a spinal nerve. You have the telencephalon. You have the diencephalon. Mesencephalon. Medencephalon. Myelencephalon. Telencephalon. Diencephalon, mesencephalon, medencephalon, myelencephalon. Telencephalon, diencephalon, mesencephalon, myelencephalon, medencephalon, myelencephalon. And that does it for the brain.